We'll get started here in a couple minutes. Deja vu, the last time I was in here.
Oh, really?
Okay, I think we'll go ahead and get started. So thank you all for coming. We'll go ahead and get started. So good afternoon and welcome everyone to today's Bohan Visiting Professor Lecture, which has graciously been supported by the School of Medicine Bohan Visiting Professor Program, the KU Alzheimer's Disease Research Center, and the Departments of Population Health, Neurology, and Biostatistics and Data Science. It's a great honor for me today to get to introduce to you our highly distinguished Bohan Visiting Professor, Dr. Amy Kind. Dr. Kind is the inaugural Associate Dean for Health Sciences and Programs at the University of Wisconsin School of Medicine and Public Health. In this role, Dr. Kind serves as Executive Director of a $400 million grant-making endowment, the Wisconsin Partnership Program. This program distributes endowment earnings of roughly $15 million annually to community programs, to faculty startup packages, and to other noble research and service endeavors. Dr. Kind directs the Center for Health Disparities and Research at Wisconsin. In fact, she is the founding director of that center, and prior to that was founding director of the Health Services and Care Research Program in the university's Department of Medicine. Dr. Kind is a professor of medicine in geriatric and gerontology with board certifications in internal medicine and in geriatrics. And she has clinical interests in Alzheimer's and related dementias, as well as geriatric transition care. She also leads the Care Research Corps for the Wisconsin Alzheimer's Disease Research Center. Dr. Kind began her studies earning her bachelor's degree in molecular biology in 1996 from the University of Wisconsin, after which she went to Washington, D.C. as a Truman Scholar and worked for the Brookings Institute. Following this, she earned her MD in 2001 from the University of Wisconsin School of Medicine. Dr. Kind completed her residency at Mass General before returning to Wisconsin for fellowships in geriatrics at the university and in older women's health at the VA. She then furthered her training to earn a PhD in population health sciences from Wisconsin, completing this degree in 2011. It's interesting here to note that Dr. Kind, or rather Dr. Kind, is a first-generation college student. Dr. Kind is well published, particularly in top-tier journals, such as those in the JAMA Network, Annals of Internal Medicine, and the New England Journal of Medicine, which published her work, Making Neighborhood Disadvantage Metrics Accessible, the Neighborhood Atlas, in 2018. Dr. Kind's Neighborhood Atlas provides census block level characterization of disadvantage, which enables researchers to study its impact on health. Disseminated freely to the public, her Neighborhood Atlas website has been accessed well over 750,000 times and counting. In addition to many other substantial leadership contributions, such as Senior Associate Journal Editor and NIH Study Section Chair, Dr. Kind has been recognized with the American Geriatric Society's Award for Outstanding Scientific Achievement for Clinical Investigation. She has been elected as a member of the American Society of Clinical Investigation, and she has served as a member on the White House Task Force on Research and Development for Technology to Support Aging Adults. This represents just a handful of a much greater list of achievements and accomplishments. Dr. Kind, we are honored by your willingness to come to Kansas City and to share your valuable time in speaking with us and also in meeting with many of our people here during your visit. We sincerely thank you for being with us here yesterday and today. With that, please join me in welcoming our distinguished speaker this afternoon, Dr. Amy Kind.
Well, thank you so much, Dr. Mencken. I'm just absolutely honored to be here. I want to thank all of the organizers, all of the sponsors, and all of the incredibly talented faculty, leadership, and staff that I've had the opportunity to meet with, to share wonderful discussions, share wonderful meals with across these last 2 days. I have been all over the Midwest, but this is my first time to Kansas City, and I'm absolutely charmed by your town. It is beautiful. Everyone that I've met has been welcoming. They've been very, very proud of the barbecue that is offered here. And I have heard all sorts of things about the history of the town, the wonderful work that's going forward here, the importance of philanthropy and of community engagement, and all that is yet to come. So you all should be incredibly proud of your institution, be incredibly proud of the work that's going forward. And certainly I have learned a lot and really enjoyed the conversations that I've been able to have. So I am here today to share with you a whirlwind tour of a particular field called Exposome study. But before I do that, let me explain this slide. This is a picture of the University of Wisconsin, and I always like showing it because I don't think many people realize that we own most of the lakefront property in Wisconsin, in Madison. So that's the university situated right between the two lakes. It leads to a little bit of craziness with the roads as they're funneling into the isthmus and out of the isthmus. But this is definitely a summer view. In winter, there's a lot more white snow everywhere. So let's see if we can get it to advance. There we go. So our outline, thank you so much, though. He was on point. Our outline today, first of all, I'm going to introduce you to this construct of the exposome, which may be new to some of you. It's an emerging area, and it is very important in terms of National Institutes of Health priorities. Next, we will focus on the social exposome in particular and its connectivity to brain health. And this is my particular area of study. And then finally, I'm going to provide you with an overview of the neighborhood study, which I'm very, very proud to say that we have many different sites across the country. And our site PI here is Dr. Mencken. So thank you very much for doing that and for carrying us forward. So first, introduction to the exposome. What is this construct? What is this word? Well, to get there, we have to start in the field of precision medicine. So I'm hoping most of you have heard of the construct of precision medicine. It traditionally involves deep phenotyping of factors that are individual to a particular person's biology. So what is it about this person that makes them unique within themselves? Typically, this is a focus on genetics, and has launched the Human Genome Project with the hope of developing new cures, new approaches, new screenings across populations. Yet we know health is an addition of not just genes and the genetics within us, but the environments in which we live. So this environmental portion is increasingly recognized as critical to producing health outcomes within single individuals and across populations. In fact, when we look at most types of the chronic conditions or those conditions that influence many sectors of our population, genetics are not the only cause. Environmental exposures are important, and importantly, they may be modifiable. So with this eye to moving precision medicine forward, to moving us into the next generation, the area of exposomal study has been increasingly an area of focus. And it is this idea that all of the environmental exposures that influence a human affect the genome and ultimately affect the expression of that genome towards health or disease. The term was first coined in 2005 by Dr. Christopher Wilde, and now it is attributable to all non-heritable factors affecting the gene expression across the lifespan. This represents a new frontier in biomedical research to complement the genome, and the NIH across the Institutes of Health, you'll notice the slide comes directly from the NIH, is looking to invest heavily within this area. So very, very important towards new pathways towards cure and discovery. The National Institutes for Occupational Safety and the Centers for Disease Control have also been very interested in this construct, and this definition comes from them. So the exposome being the measure of all of the exposures of an individual in a lifetime and how those exposures relate to health. So again, this idea of that environmental piece. Coming out of the field of neurobiology in particular, this study was published in Science in 2020, thinking of the exposome as all of those factors external to the biological human. But they're very diverse indeed, and they can include exosystems, lifestyle factors, social factors, and physical chemical factors, and range from microbiome studies, which are technically that microbiome is external to the biological human, although it may be within the gut, of course, all the way through to structural inequities and the core of many of the inequities that we see within our society and others. So when we think about challenges to examining the exposome as an entire field, each of these areas could be its own department. This is how broad these things are, but these are the major challenges. The first is, how do you quantify these exposures? How do you take exposures across an entire life course and measure those in a rigorous, harmonizable, and reproducible manner to be able to apply to studies? Next, how do you take those measures and link them to biology? So we can look at social biological mechanisms, particularly when many of our biological resources and infrastructure have been de-identified or otherwise unlinked to social factors. So how do you add those back? How do you think about that? And finally, since the exposome is theoretically modifiable, how do we link this research to action to change the pathway of health across populations? So in the field of social exposome and brain health, I hope to touch on each of these as just an example of how our group and our partnering collaborators have approached this particular space. So what is the social exposome? So it's one portion of this larger construct of exposome, but it is an area that can be quantified, studied, and modified. And many measures of the social exposome are already available from our colleagues in sociology, in demography, and through decades of research within the social sciences. There are many, many areas that have already been discovered and put a lot of wonderful thought and rigorous research into. But these measures of the social exposome often reflect structural inequities in any society, be it in the United States, in other countries across the world. There have been historical reasons or current reasons, certain policies, certain laws that have led to unequal distribution or access to resources that ultimately affects health and affects the way a human interacts with their environment. An important thing about the social exposome is to know that it's not just a proxy for individual level social factors. An individual person will have characteristics about them, and in addition, they live in an environment that in and of itself has its own characteristics. And from many, many decades of research in many other fields, and certainly from our own teams as well, we can show almost full independence in terms of the impact on health from individual level social factors as opposed to exposomic level social factors. They are sometimes correlated, but they both are important in terms of health. So you don't use one as a proxy for the other. You actually want to include all of these if you can. And of course, as I mentioned, there are entire departments dedicated to this. They will not be labeled as social exposome, but they've been thinking about social factors and the rigorous quantification of those. So we can work with those partners to move health forward. My particular interest is in health disparities. And increasingly, the health disparities research field is embracing the social exposome approach as a pathway towards discovery and action. And we can see some of that through some of the health disparities frameworks that have been published within National Institutes on Health institutes. This one happens to come from the National Institutes on Aging. And for those of you who are interested in health disparities research, I would encourage you to look up this paper by Carl Hill and colleagues that was published in Ethnicity and Disease in 2015. This forms the framework also for the accepted health disparities model that has gone forward from the National Institutes of Minority Health and Health Disparities. And the Minority Health Institute is the one that often will help other institutes to figure out how they move forward in this space. But fundamentally, as we think about this model, we can simplify it into these 4 domains. So when we think about these domains, we can think about them as environmental, sociocultural, behavioral, and biological. And these do not work in silos. They interact with each other across an entire life course to either produce health or disease. So as we think about studying health disparities, we think about intervening on populations and trying to improve health outcomes. It's important for us to recognize that there is opportunity across all of these domains to build multidisciplinary teams that can come together in really unique and innovative ways towards new discoveries, new pathways of clinical intervention, and new social policy. But again, that blue bucket, that environmental bucket, we can think about the exposome right there. So there are many examples of structural inequities across our country and others, and I will show you just one. Do any of you know, just by a show of hands, what this is a map of? Yes. So yes, sir. This is a map of redlining. So this is a historic map from the city of Philadelphia. This happens to be Philadelphia. I could have pulled lots of different cities. This one just happens to be Philly from 1930s. And this was a map created by bankers. So this was a map created by these lenders in federal spaces or sometimes private spaces. And you can see that different areas are marked out in this map with different colors. They've used a pen to color different neighborhoods, either blue, yellow, red, or there's no color. And on the legend, you can see that the red areas are denoted as hazardous. So what this would mean is if back in this time, when you went to your local bank or you went to a lender, they would pull out this map and say, tell me your address, tell me where you lived. And for those individuals who lived in different areas on this map, they would have different access and ability to get those loans. So if you lived in one of those red areas, you were more often turned down and you didn't have access to different types of loans to start a business, to invest in your community, to buy a home, to be able to move forward. Now, most of these neighborhoods this was primarily an urban phenomena. Most of these neighborhoods were African-American neighborhoods, other racial and ethnic minority neighborhoods, or religious minority neighborhoods. And to this day, in many areas, we can still see the outline of these red line practices in disparities metrics that we can build now. So these policies have echoed forward to today. In some ways, some of these policies still have legacies into today. And there are obviously other examples of structural inequities, systematic racism, systematic other types of isms that go forward, but they impact the metrics that we see on neighborhood disadvantage, like the area deprivation index that I'll show you soon, even to this day. So the exposome really is, as we think about the exposome, we think about these neighborhood measures, it is a measure of distribution of resources. It is a measure, a way to think about structural inequity, and it's a way to think about linking some of the precision medicine approaches that have been taken to health disparities research to lead to new discovery. So this issue of quantification, how do we begin to quantify the social exposome and study it in a way that's rigorous and harmonizable across an entire country, across multiple populations? And how do we ensure that different groups can have access to these measures? This has been an area of research of mine for almost 2 decades. In thinking about multi-domain indices of disadvantage, working with those indices and developing new indices and also refining existing indices for use across broad populations. Our work has been funded by the National Institutes of Minority Health and Health Disparities and the National Institutes on Aging, but has focused over the course of the last decade or so on this measure called the Area Deprivation Index, or ADI. The ADI is a multi-domain indices of disadvantage that includes 17 different measures of social determinants of health that are pulled together using a standard mathematical approach across very small population-sensitive areas, these census block groups. And what it allows us to do is to differentiate areas of challenge in the social exposome domain, areas of adverse social exposome, in a very precise way that minimizes measurement instability. This is a rank-based score. It's time concordant. So we've built these metrics all the way back to the early 1900s in some work I'll show you in a moment. And it's been strongly, strongly linked to health. And you may ask, why is the geography so important, these geographic levels? And I like showing this example of Milwaukee. So Milwaukee County is one of the most challenged counties in terms of adverse health outcomes in the state of Wisconsin. It has some of the worst maternal fetal mortality rates in the entire country. And when you look at Milwaukee at a county level, this whole map is one county. You look at it just at a county level and you see that it's highly challenged, highly disadvantaged. However, when we use this much more precise geospatial measure that goes down to the census block group, what you see is a county with many different patterns. You see blue, you see red, you see yellow. Now, the red areas are areas that score highest on this metric, this ADI. They're the areas that are most disadvantaged. The blue areas are the areas that score lowest on the ADI. They're the areas that are least disadvantaged. You can think of them as very wealthy neighborhoods. And I'd like to note that the wealthiest neighborhoods in Milwaukee are right along that lakeshore. How about that? That is Lake Michigan. And then we have an inner city core that is incredibly high in its ADI scores, these red areas. Those red areas correspond very directly to areas that experience redlining. And they are areas with the greatest adverse health outcomes. So what this helps us see with this more precise measure is that Milwaukee County is actually an incredibly diverse county in terms of its resources, with areas of the greatest wealth, some of the richest neighborhoods within the state of Wisconsin, as well as some of the poorest, often abutting each other, one right next to each other. So using this much more precise approach, it allows us to think about resource targeting, it allows us to think about risk assessment, and it allows us to think about the social factors that interact with biology from a much more precise lens. And so because of this much more precise level has been really important for us to have made the strides forward and for the field to make the strides forward that I'll show you in a moment. When we first started building these metrics, we were asked for them so often that we decided to build a data democratization tool called the Neighborhood Atlas. We launched that tool in 2018 with the goal that it would be targeting non-research audiences. And to this day, our most common users are coming from community groups, policy groups, and health systems. And it has been designed to be a tool that is meant to be very intuitive. It's meant to be something that you can interact with maps just like you do on your smartphone. You can use a smartphone, you can use the Atlas to make meaning of these 39 million data points that otherwise you wouldn't have access to if you didn't have an advanced data science degree. To date, we are getting about 1,000 downloads weekly of the data on the Neighborhood Atlas. Most of these are going to non-research groups. For researchers, then there have been over 1,200 papers that have been published since 2019, and over 800 of those were just in the last year. So the tempo of use continues to increase, and I'll show you examples of how it's been used in state and federal policy since 2022, but it has become the cornerstone of health equity measurement across a number of policy approaches. So when you work with the atlas, you can immediately see maps. This is obviously Florida. You see areas that are blue. These are areas of low ADI, the more wealthy areas, as opposed to red areas. The deep red are the highest ADI areas. You will also note that the colors are in different sizes. And this is because the census block groups that they're anchored to are all about 1,500 people. And in rural areas, they have to be geographically larger to capture 1,500 people versus urban areas that can be very small indeed when there's population density. Here's Missouri. I thought I'd throw up a picture of Missouri here. If you work with the atlas, you can Google neighborhood atlas and you can map anything. You can map your home. Lots of people map their home. You can map the areas that you care a lot about, but you can also zoom in. And we have an entire team of geographers that helps us with the geographic layering here to help particularly our community members and other users to make meaning of these maps. When we look across the United States, the highest ADI neighborhoods tend to be in inner city areas. Particularly, we tend to see this pattern like you see in Milwaukee, where you have an inner city core that's incredibly red and high ADI surrounded by a ring of incredibly wealthy suburbs. And that pattern is repeated again and again across the United States. Additionally, Native American and tribal areas tend to show as highly disadvantaged on this metric, and rural areas like West Virginia. Note the southeastern United States and some of these areas that have a disproportionate lessening of mortality, higher risk of stroke, higher risk of cardiovascular disease. They also show up as high ADI areas. In terms of the social exposome and neighborhood disadvantage by ADI, I could give you, there's been obviously thousands of papers, most that have not come from our group, but since we're going to be talking about brain health, I'll give you some highlights there. It has been linked to an increased risk of dementia diagnosis. This was a study that came out of Christine Yaffe's group, was published in JAMA Neurology just last year using veteran affairs data, showing veterans who are living in high ADI areas who otherwise had access to healthcare were diagnosed at much higher rates of clinical dementia diagnoses. Additionally, there have been studies that show individuals who live in these areas have greater risk of cognitive decline and are more apt to have positive AD biomarkers, particularly tau. There have been a number of studies now showing that these types of multi-domain indices of disadvantage link tightly to epigenetic age. And this finding now has been replicated within the United States and outside of the United States using metrics that are harmonizable to the ADI in other countries, including out of the UK Biobank in the UK and out of Australia, in which when researchers look at the biological clocks that can be found by the telomeres and the epigenetic signatures, they find that individuals who are living in these highly disadvantaged contexts ultimately are older in terms of their biological clocks than their counterparts in more wealthy areas. Living in a highly disadvantaged neighborhood, a high ADI neighborhood links to increased risk of hospitalization, greater care costs, poor care quality and access. It's marked to increased risk of post-surgical outcomes, adverse outcomes, and complications like delirium. It's been linked to increased cardiovascular disease risk, cancer, diabetes, obesity, the list goes on. And out of a study with Yale, out of a longstanding cohort study, Tom Gill and colleagues showed that individuals who live in these highly disadvantaged areas by ADI live shorter lives, and the last years of their life are more disabled. So they're more apt to need in-home services, nursing home services, and experience disabilities overall. I won't go through the entire literature in this area, but just safe to say there's a lot. There's a lot. So depending upon your area of interest, be it diabetes, cardiovascular disease, cancer, Alzheimer's, there's all sorts of studies that have been published. There also are increasingly interventions that are using the ADI to target resources and research groups that are using it in order to characterize their cohorts in ways that allow for this lens of ADI to be interpreted, and also to use these geospatial markers as a way to target recruitments preferentially. In the brain health space, it's leveraged across many, many studies, including I'm working right now with the Alzheimer's Clinical Trials Consortium and their recruitment efforts as they think about differential secondary analyses of existing data to look at differential response to certain treatments, as well as recruitment, outreach planning, and strategic planning. working with a number of pharma companies for the same reason. There's a lot of interest in trying to decide where the Pfizers and the Mercks and these other types of groups establish clinical research outreach efforts, and increasingly moving beyond kind of traditional academic foci into communities. This ability to think about secondary analysis of data. Sometimes you will have populations that respond better or worse to certain interventions, particularly health systems and clinical interventions that may not be fully tailored to the population's needs, and that can be learned more using these data. And then often these groups are collecting ADIs at the point of enrollment, or they link by geocoding from the addresses they have, so from a cross-sectional standpoint. And so with this, they turn to the atlas, they use it as a tool, or they download the data and build their own platforms to do that automatic linkage. There's been some really interesting work out of the adolescent brain cognitive development study that suggests that children who are growing up in these areas of high disadvantage have different connectivity amongst their early brain structures, particularly between their amygdala and their frontal lobes. So even the kids of the brains of these little kids, as young as 2, are looking different for these children that are growing up in the areas of red on the map. Additionally, federal policy groups use our metrics, as I mentioned, for their health equity benchmarks. And I could give you many examples, but I'll show you this one since we're talking about brain health. The new CMS guide dementia care model, which I understand will be rolling out here as one of its sites, does use the ADI as their health equity adjustment. So what this means is if you are caring for populations that are coming from those red neighborhoods on the maps, you're going to get more money. You're going to get a bonus. And that money is meant to target social services. It's meant to target social determinants of health. And ideally, it's meant to be those types of needs that the community recognizes. So you have, through this model, you would have the community at the table. They would tell you what their needs are, and then the additional funds would be used for those purposes. Obviously, the needs in a place like Kansas City are going to be different than the needs in Jackson, Mississippi, are going to be different than Sioux Falls, South Dakota. So having this ability to tailor with the community input is important. But the ADI itself is used as the economic parameter for adjustment. Additionally, we have many, many examples at the local level. Florida Medicaid, many Medicaid programs are using ADI to target resources and assessments, but I thought I would share this one because it has impact on brain health. The Milwaukee Waterworks has a problem with lead. It's similar to Flint, and Milwaukee is in many ways actually has a greater burden of lead poisoning than Flint, Michigan. So they have this need to do a whole host of lead mitigation efforts that are incredibly expensive, and the city can't afford to do them all at one time. So the philosophical question is, how do you target? How do you prioritize? Where do you send the backhoes? Where do you send the new tubing? And they have decided, this was an article that just was from May of 2023, to adopt the area deprivation index as a way for them to target and prioritize neighborhood by neighborhood for the lead pipe mitigation efforts. So you can see this came from a news article talking about this program and the old lead pipes on the top that are coming out, and the new ones, the new copper lines that are going in. This has been so popular with the people of Milwaukee. And of course, popularity means, you know, like the politicians get voted in and things, that they're talking about using this philosophy for a whole host of other programs in Milwaukee now, which is great. In addition, I mentioned the NIH is incredibly interested in this area of exposome study, not just the ADI, but all of the other areas of exposome. So I am on the advisory board now for the National Institutes of Neurologic Diseases of Stroke, NINDS, and they just about 3 months ago launched this brand new office called One Talks, and it is all about the neuroexposome. Their intent is to fund a large array of research that focuses on exposome and brain health and moves the field forward in a whole host of ways. So they are bringing together diverse researchers, everything from social exposome that I just talked about, all the way through to pollution, microbiome, bringing us all together to think about how these features interact and ultimately influence brain health and neurodegeneration. In some of our own work, when we started looking at some of these questions about how the social exposome might link to the brain to result in associations with different types of health outcomes. We started with our own cohort at the University of Wisconsin. We happen to have one of the longest standing longitudinal cohorts of brain health called the Wisconsin Registry for Alzheimer's Prevention, or WRAP. It's led by PI Sterling Johnson, who now, for those of you in this space, know Sterling is also the PI of the Big Clarity Grant that is rolling out. But this is a cohort of individuals in midlife who are not cognitively impaired, and they have been followed over the course of decades, about 14 years, and undergo repeated in-depth cognitive assessment, biosample acquisition, including CSF and blood, PET scans, and MRI scans. So we were working, this was work by an MD PhD student. So I met a bunch of MD PhD students, and how about this? Jack, before he graduated, got his paper published in JAMA Neurology. Now he's doing a psychiatry residency at UCSD. But this work, as it went forward, the question was, in this cognitively normal cohort, were there differences by volumetric MRI between those that lived in the most disadvantaged areas by ADI versus those in the least? And what you see here are two of the findings from this paper. The first is that there was a finding that total brain tissue volume, the total volume of the brains, were different from across by ADI for those living in the most disadvantaged ADI neighborhoods, in that the brains were smaller. And in addition, hippocampal volume was also different. Hippocampal volume was smaller. Now, it was interesting because the student took this to the next level, and we worked with him on mediation modeling to try to see if we could explain these differences. And the total brain tissue difference could be almost entirely explained by cardiovascular risk factors: diabetes, smoking, cardiovascular disease. So that was interesting. And certainly we know those features, smoking, cardiovascular disease, diabetes, these are things that happen more often if you're living in a highly disadvantaged context, going along with obesity and things like that. But that hippocampal volume difference could not be explained by those factors at all. And it does raise question whether there are different physiological pathways going on, perhaps even in early life, that are leading to differences in volume and exposure. But of course, this is a cross-sectional study. This was just at one point in time. So the next question really is, could we look longitudinally? Could we look over a longer period of time? And in this study, in this same cohort, individuals were compared to themselves over the length of time that they have been in the study, most for about 10 years. And what we found is that the individuals who are living in these more disadvantaged contexts not only could be seen as having differences in their cross-sectional brain volume, but we could show that their pace of cortical thinning was much more rapid. So when compared to themselves, they had an accelerated degeneration in their Alzheimer's affected regions. And this was strongly associated with cognitive decline over that period. So these individuals had brains that were shrinking more quickly than their counterparts in other areas. And again, this is an association, but as a marker of risk, it is of great interest. As a result of these two papers being published, and again, this was just 2020 and 2021, the Alzheimer's field has embraced this ADI and has now published dozens of papers replicating these findings and showing, extending upon them in many ways. Our next question, though, is in the Alzheimer's field, so much of what we do requires postmortem tissue because we don't always have great markers for disease in life. We're starting to develop new ones, but there's lots of diseases that affect the brain that we can only see at the point of death when we look at the brain under a microscope. So the question really was, could we add back social factors to brain tissue for individuals who had donated their brains to science with the hope, this is such a huge gift of these individuals, and they're hoping for cure, they're hoping for new discovery. And so they and their families have given this very important part of themselves at the point of death. But for most of these individuals, their tissues have been in brain banks across the country. You have one here. There are brain banks all over the place. And these tissues tend to be very biologically characterized. They're really in depth. We know the genes, we know the omics, we know all this biological stuff. But often the social factors are lost. And that has just been the pattern of the way these things have been archived for many decades. So we wanted to add back social factors and we wanted to see if we could do it. This was really just a feasibility study to start with. And so we partnered with, we used our own brain bank, obviously, but then we partnered with University of California, San Diego, because they have one of the oldest brain banks in the country, one of the biggest. And we saw whether we could add back social factors for individuals who had died from 1993 to 2016. The reason for 1993 is that prior to that time, UCSD's records were in a bunch of manila paper files. And so we wanted to start with the electronic records where we had them. We pulled their autopsy reports and the autopsy reports for all of these individuals, and we looked at the address that was on the autopsy reports for where these individuals were living at their point of death. We geocoded that in a time concordant way and linked it to the appropriate area deprivation index, ADI, for that time when the patient died. Remember, ADI is time sensitive, so we have to build each of these things going back in time. And what we found, first of all, our biggest finding and our planned finding was that it was doable. We could do this. It was feasible, which what an odd thing to do, right? And yet we could do it. We did not expect to see, but did find a strong association for living in the highest ADI decile at the point of death. Those individuals had a marked increased odds of AD neuropathology, almost in a dose-dependent fashion, which was fascinating. It was the first time these tissues had been linked to social factors in a way that could combine these two domains for learning. But of course, life course becomes important. And when I introduced the concept of Exposome to you, we talked about different types of exposures across an entire life course. So how can we take the data that I just showed you and extend it back to the point of birth? How can we think of those risks for brain health, including dosage and timing of different exposures and how they may link to adverse health outcomes? We'll use the brain as an example, but the methods that I'm showing you could be applied to almost any disease state. And this raises a question whether there are critical windows. What if it is childhood that is writing the future for where someone experiences diseases and how they experience diseases in their 70s and 80s? What if it is those critical windows that really matter the most? And this is where we're just trying to take baby steps to start to build the infrastructure to answer these types of questions. It's incredibly heavy lift. And this brings me to the neighborhood study. This is the study that was born from the preliminary data that I just showed you with this question of longitudinality at its core. It is when it was originally awarded, it was a $28 million R01, which now has been expanded with some supplements to be larger. But it's designed to examine the impact, mediators, and moderators of the life course exposome on Alzheimer's disease and related dimensions, pathologic features, vascular burden, and cognitive decline. Each of those blue states there, not the light blue states, but the dark blue states, are ones that we're targeting for the study. And you can see all of our different study sites here, including the University of Kansas. Thank you very much, Jonathan, who is our lead here. And you can see the other sites, though. We have good coverage across the country, and overall, there are over 60 key personnel in this study, so very, very big study. Our goal is to characterize the life course of individuals in a couple of ways. First of all, for our over 10,000 brain bank decedents, we're using something called residential history tracking, which I will show you in just a moment in greater detail because it's very novel. And for the living clinical core participants, the way of assessing residential history is somewhat more straightforward in that we use surveys. And we ask individuals where they've lived across their lifetime. The technological lift to actually geocode back in time is not small. And that is one of the biggest challenges to the study, as is data share of protected health information and addresses. Our Center for Health Disparities Research specializes in just this. So we have advanced GIS capabilities. I have an entire team of PhD geographers and master level geographers that do a lot of customized geoanalytics. You can, of course, buy geocoding tools off the shelves, but those are for the modern era. They don't really help you for 1910, for instance. So to go back that far, and our earliest individual in the cohort was born in 1896. So to be able to go back that far requires quite a bit of technological lift. We also do this public data tracing for decedent residential history, and we have a large health equity policy group, but we'll focus on that middle box right now. So how do we get a residential history from someone who has passed on? This is very innovative, but in one way, it's innovative for bioscience, but it's not innovative for history, because what we are doing is basically doing historical archival research, like a history department would do, like countless probably PhD students here in your history department do as they trace back whatever their question is of note. But we use public data tracing for each of the individuals who have donated their brain, treating them and thinking about this precious, precious gift and characterizing their social history all the way back to the point of birth through the public archive. It is adding back, I would argue, some of the social humanity to the bioscience that has already been done on these brains. And it gives us an opportunity to really shine new light on these findings. So this gives you a sense of the birth date and death date of our samples. All of these individuals are deceased. As you might imagine, you cannot live without a brain. So our sources for records, I hire countless numbers of historians on the team. They search through city directories, census, military, pension records, marriage records, death records, and probate records, and also newspaper articles. And the obituaries are a very rich source of information about what someone has done in their life. And using this approach, we have two archivists that work independently to ensure rigor. This uses a lot of the same techniques that one might use in old school abstraction research that used to be done before. Now we pull everything electronically from the EHR. But using these approaches, we are able to discover 73% of all person years are discoverable. So this is a very high level of discovery in terms of our residential history and the addresses. Each of these documents has an address, but we create the archive for future query as well. And for a number of living participants that are participating in our survey, we asked them if they would be okay if we did a public tracing on them and compared the two results just to validate this approach. It's very high concordance. In fact, the public data tracing seems to be better. You know, people are having recall bias about kind of the name of their street and things, and we can find these things. So it's over 98% concordance. So in terms of some of these initial findings that'll be going forward with the neighborhood study, some of you in this room know some about them. Since this is a public forum, I cannot out them yet because we have submitted them for presentation at the 2024 Alzheimer's Association International Conference. But for those of you, once we're able to present at that conference, we will be able to share information on the cross-sectional distribution of ADI and these brain bank cohorts at the time of death, and that association with neuropathology. And there are some very strong associations that we're finding. So some new directions that our team will be going. I thought I'd share some of this with you. There's great interest in military history, in this idea of different exposures that veterans may have experienced when they were deployed, when they were active in the armed services, including different types of toxin exposures, different types of battle stress, different types of injuries that they may have encountered while serving their country. And so I have a assistant professor that is with our center who is taking the lead on this in linking military history to neuropathology within these cohorts. To the best of my knowledge, this will be the first time that such a repository will have been built, or such linkages will have been built, and hopefully gives us an opportunity to really dive deep into some mechanisms, or at least trigger some hypotheses that can allow for additional study. As a first step, this individual has worked with this same preliminary brain bank cohort that I mentioned from University of California, San Diego and UW. And what we were able to do is demonstrate those individuals who are noted as having a military history within their public archival tracing. It is very typical when someone writes an obituary that they will highlight someone being a veteran. So that was noted, not just from the obituaries, but also from the archival documents that could be found. And using that as an indicator variable, we were able to find that UCSD's sample in particular had over a third of their individuals as having military background, most of whom were in the Navy. If you know anything about San Diego, it's a big Navy port. But this gives us an opportunity to really kind of think more about how military service may be associated with some of these late life changes. So this is an example of some of the public records that get found in this type of research. These are being shared with permission of the family. They are very proud of this and would like you all to see this. So I am sharing this with you for this particular individual. So this was someone, for instance, in which across the public data tracing, there was a newspaper article that was found. This was from the 1940s. About this man coming home after World War II, after 18 months of duty in the Pacific Theater. And then what you're seeing on the other side of the screen is his application for World War II compensation. These are documents that our team is pulling from. You can see these are handwritten documents. They tend to be messy. Sometimes the words are hard to see. So right now it does take a human being to really look at them and extract the information. We would love to get to a place where we can automate this process and be able to pull information directly from these archives. But right now the archives are in so many different places and it takes so long to do one of these tracings that for the present time, it's a human endeavor. But using this approach, Dr. Powell published just a few months ago, actually, in Alzheimer's and dementia, this strong association between any military service and Alzheimer's disease neuropathology at autopsy, showing that amyloid burden was increased by 26% and tau by 10%. He has since gone on to characterize the different branches of service on the basis of the obituary and ultimately finding that individuals right now who were in frontline positions seem to have a higher burden of disease. So more to be done here, much, much more to be done. And obviously, we are hopeful that this kind of research will have important ramifications for our veterans even now in terms of eligibility for services and additional kind of disability compensation and things of that nature. So, and that there will be new opportunities and windows opened up to try to prevent this within our armed services as well. We also are, as I mentioned, are very interested in other sorts of geospatial characterizations. I showed you those redlining maps. Turning those color-coded maps into data that can be mapped onto today's census block group is a little tricky because those maps don't really match today's census block groups. Things have changed quite a bit. So our team is starting to do some of this cross-linkage. There is a group that publishes these Hulk maps already and provides a lot of tools that can help us towards this. So this is something that we are interested in going forward for. One of the things about the research we do is it crosses coast to coast in the United States. And the Hulk maps right now are just available for those gray spots on the map. They're just kind of urban areas and they're somewhat spotty, but we are starting to look into this and seeing if there are some interesting additional windows that we can provide. Through all of this, and since this is you all are primarily an academic, obvious audience and a research audience, I also wanted to share what I found is one of the biggest challenges to this type of work. Of course, there's the technical piece, but there's also the legal piece. And this is very challenging. So how do we balance this protection of protected health information? I'm dealing with addresses, right? And geocoding and all of this is PHI while facilitating a broad science, while moving the science forward. So it involves this balance. The definition of protected health identifiers under HIPAA is that they're all geographic subdivisions smaller than a state, including street address, city, county, precinct, zip code, and their equivalent geocodes. ADI is not. ADI is not a protected health identifier. So what we've done is build infrastructure to allow for that linkage. And then what we share is not the geocodes or the PHI, but it's the exposure variables like ADI or military service or whatever the case might be. So that's been our approach. But the pathway towards this for the neighborhood study was considerable because I spent more time than I can count talking to lawyers at these 21 sites developing detailed and customized data use agreements with each site. HIPAA is different with every state. So this was a substantial undertaking, but now we have all of this in place. So as we work with, for instance, the University of Kansas, we have a pathway by which to safely and securely transfer data, store data, process data. And in our next chapter for the neighborhood study, we are working on cloud-based systems that will allow our collaborators in many places resources to come together under those cloud platforms to work with us. For now, we will be sharing out the de-identified limited data sets, but that is the vision. So this takes a lot. We have many biorepositories within the Alzheimer's space that have no interest in managing PHI because it's such a massive, massive lift, including NAC and AMP-AD. Many of these are kind of the alphabet soup of the Alzheimer's field. So if those of you that are in Alzheimer's will know what these things mean, but they're basically national resources that either curate genetic data or they curate biosamples, or they do something that's very specific. And what we are proposing to do is developing safe pathways by which to add on measures like ADI. So our basic science colleagues can really start thinking about some of these social interactions. So in conclusion, I'll stop there. Hopefully I've taught you a little bit about the exposome, and it's an emerging construct. It's really, really important in the field of health disparities and human health research. And it's theorized to interact with biological mechanisms to influence health, including brain health. The social exposome is actionable, it's measurable, it is used across policy now. Whenever I talk to policymakers, non-research audiences about the ADI, I frame it as a social exposome measure, and I show them how it's linked in science to things like neuropathology, and it has been very powerful in that way. And the neighborhood atlas is available for all of you. It's free, it's available. We ask you to register because if we have to update the measures or we're doing things, we'll send you emails, but there's no cost. You can use the data. And then finally, the neighborhood study is our current big research initiative. And we're really, really excited about the infrastructure that it's establishing to facilitate more study. So I'll stop there, but I have to thank our many, many, many researchers that have been involved with this study, including Dr. Menken, Dr. Morris, Dr. Lepping, who are all here at University of Kansas, leading the sites here. Obviously, we couldn't do this without the patients and the participants that have given so much of themselves, their time, their blood, their brains, in order to try to find a cure for Alzheimer's disease and other neurodegenerative disorders. And then, of course, the National Institutes on Aging. This is the list of funding. It goes very, very long. To do stuff like this takes a lot of partnership, and it could never be done by one person. So I also have to thank the entire team at the Center for Health Disparities Research. What I showed you today is the product of many, many, many people coming together in a mission-aligned way in order to try to improve human health, and particularly brain health. So thank you all very much, and I'll take questions.
So I think we've got some tech support for the questions. So if you have questions, we'll get you a microphone. So questions. Is this on? Okay, terrific.
Oh, that's so cool.
This is super exciting. Thank you so much for your talk.
Thanks.
How does gentrification affect the ADI?
So that's a great question. And the place we see it, we can see it very well in Baltimore, for instance, the Inner Harbor versus the rest of the city. And so our the parent data that forms the ADI is drawn from something called the American Community Survey. They're 5-year estimates that come together for this block group. And they do have a little bit of statistical variability across time, but they do tend to change with time. It gets very complicated in terms of response rates and how the census collects that data, but we do see changes. When we map ADIs across time, prior to about 2010, we used long form census data, which were collected every 10 years. So we see changes across those 10-year periods. And then in the more modern era, we can look at 1 to 5 year changes. What we see is in places like Baltimore, the Inner Harbor changing. We do see places like Detroit. Detroit in the 1970s looks very, very different than the Detroit of the early 2000s, which looks different than the Detroit of today. Actually, there's been this kind of change over time in terms of the relative rankings. So we do get that across time, but our time frame of precision will depend upon census's collection patterns. So every 10 years in the previous period and every more frequently in the more modern era.
Thank you.
Yep. Now, let me also say there are very detailed measures of gentrification that are available in the social sciences. ADI is not technically a measure of gentrification. We do see gentrification a little bit, but if you are interested in that construct as well, there are excellent, very rigorous measures that are available too.
Thank you.
So, yeah.
All right, we do have a question in the chat. Someone is asking if any of the ADI research has looked at cognitive changes after cancer treatment, and if there's also a connection there.
So I'm not aware of that particular question being answered, but I do have to admit, I can't keep up with all the literature. There's just been, it's almost 1,000 papers just in 2 years. So I get the citation alerts and I look at the titles, but I don't always dig in. So I think that would be an area of great interest. I can say from my memory, in the cancer space, there's been a lot of interest in post-surgical outcomes and how well patients do, how long they survive, what types of complications they might have. And that has been of great interest. A lot of the cancer, I've seen in the cancer surgical space, there's been a lot of use of the ADI. And again, I think our surgeons are so interested in who's going to do well or poorly after a surgery. They're often thinking very much about risk prediction so that they can counsel patients and work very closely with them. But I cannot recall something specifically on cognition in the post-diagnosis cancer state. I think that would be an area of really important research, and who for that individual, I would encourage them to follow that. There are hundreds of questions to be asked in this domain, and the NIH is very interested. So it's a good area to think about. Yeah.
I'm wondering about people with a lived experience of incarceration
Oh, yes.
and if there's tools in the ADI model to account for that for folks.
I think that's an excellent question. I would say in the health space, incarceration research is in its infancy. I think there's a lot more to be done. In the ADI itself does not account for incarceration. And as a matter of fact, prisons do not submit census data. So prisoners do not fill out the census forms. So sometimes you will look at a city and there's this like gray area. And typically those are areas that have been suppressed on our ADI maps because of maybe a large institution that doesn't, like a hospital that wouldn't submit census forms, but also prisons and military bases fall into these categories. So we know where the prisons are located, but we don't always know who was in prison. And thus far in the neighborhood study, we have not been collecting that information. I can say though, I have a junior faculty member who is studying the care and treatment of prisoners in the hospital. So individuals who get sick enough in prison that they have to transfer into the hospital. And at the University of Wisconsin, we're the main provider for the Wisconsin Department of Justice System. So we have lots of prisoners that come through and receive healthcare services from our hospital. And so she is approaching this and trying to dig into the data, and the data are hard to come by, first of all. Secondly, though, there's so much to be done. For those of you who know what delirium is, it's this brain complication that can happen when someone gets sick or during a post-operative period. And her goal is to try to improve delirium detection and care in that population. So often these prisoners are, they do not always receive the care that we would expect an older adult to receive to prevent delirium. For instance, it's very typical for a care pathway to include 24/7 shackling to a bed or other things, which is restraints are one of the worst things you can do for brain health. So it's things like this. And obviously these issues are super complicated, but it is an area of great interest to our center, but the data has not yet kind of been there to do all of the, answer all the questions that are of interest. I think it is an area of interest as well, and very obviously lots of needed study is needed. The data resources are tricky. Other questions?
And I think we're actually running up on time, so let's please join me again in thanking our.
Thank you all.